Replication is the duplication of DNA. In eukaryotic cells, this occurs well before mitosis and cell division. In prokaryotes, like bacteria, replication is concurrent with growth and cell division. The structure of DNA lends itself to hypotheses for replication that were essentially correct. Recall that Griffith discovered that live cells could pick up genetic information from dead ones, a process he called transformation. Avery and company quickly showed that the transforming principle or transforming molecule was DNA, and then Hershey and Chase showed that the genetic molecule of viruses was also DNA. When Watson and Crick unraveled the structure of DNA and proposed the double helix, the complementary relationship of the antiparallel strands of DNA led them to propose that DNA replication was in fact semi-conservative. This was tested formally by Messelson and Stahl and shown to be correct. There were three possible models for how a double helix could replicate. All involve the separation of the complementary DNA strands. At the left is conservative replication, in which the parental double strand is indeed conserved. In the middle is semi-conservative replication, in which parental strands each combine with a new daughter strand. Messelson and Stahl had access to nitrates made with heavy nitrogen, or N15. They grew cultures of E. coli in media containing the heavy nitrogen, and after several generations, all of the DNA in the cells was heavy DNA, shown as blue strands to the right here. Then, cells were transferred to medium containing normal or light nitrogen, N14, and allowed to grow for one generation. For each replication model, they could predict what kind of DNA would be replicated, as illustrated here. They would see either 100% middleweight DNA or two distinct kinds of DNA in equal proportions. And here are those predictions. Messelson and Stahl did the experiment and extracted DNA from the first generation of cells. If the cells produced a 50-50 mix of heavy and light nitrogen double helices, they would be able to separate them by a technique called density gradient centrifugation, a fact that they had previously demonstrated. The cells, in fact, contained 100% middleweight DNA, that is, an equal combination of light and heavy nitrogen-labeled DNA that behaved as if it was all of one density. So that eliminated the conservative model of replication. That left semi-conservative and dispersive models of replication. The dispersive model was eliminated by carrying this experiment out to a second generation of growth on the light nitrogen isotope. Now, can you explain why?